In this lecture, I am going to talk about anti-isotype antibody, anti-allotype antibody, and anti-idiotype antibody. This anti-isotype antibody, actually anti means against a isotype of antibody. What is isotype? You have to first know. Isotype means class of antibody. There are 5 isotype or class of antibody. These are designated as immunoglobulin G2. You can remember these 5 antibody as a GAMED, GAMED. It can be written as IG, G, IG, A, and so on. So these 5 are actually isotype. This isotype of antibody actually encoded in our gene in different gene segment. Let's assume this is a alpha, this is a gamma, this is epsilon. So this is a gene region. I want to mention that antibody is composed of 2 segments, 2 chain. One is called heavy chain. This is heavy chain. Another is called light chain. This both heavy and light chain consist of 2 regions. One is called variable light and constant light, and in case of heavy chain it is called variable heavy and constant heavy 1, constant heavy 2, and constant heavy 3. So this is the constant region and this is variable region. Isotype of antibody differ in the constant region of heavy chain. So they differ in this region, only this region. So I can write that isotype of antibody differ in the constant of heavy chain domain. So anti-isotype antibody means this antibody is against a particular CH domain of isotype. Let's assume this is a anti-isotype antibody. If this antibody recognize a particular constant heavy domain like IgG, then this anti-isotype antibody, this is an anti-isotype antibody, cannot recognize other type of like A, M, E and D. Anti-isotype antibody means a antibody that is against a particular type of isotype. And in this case I want to mention the light chain of antibody also can be of 2 types called kappa and lambda. And this anti-isotype antibody can also recognize the constant light region of kappa or lambda. So this anti-isotype can also recognize constant light. If this antibody recognizes Cl, means constant light of kappa, then it cannot recognize the lambda, or vice versa is true. A anti-isotype antibody bind with constant region, means C region of heavy and light chain. Next is how this anti-isotype antibody is generated. Where from this antibody is generated? Anti-isotype antibody. So the answer is, if there is 2 species like A and B, and if I transfer antibody from species A to B or species B to A, these 2 species will develop anti-isotype antibody. Different species, you have to remember. So this is all about anti-isotype antibody. The next is anti-allotype antibody. So this antibody is also against a allotype of antibody. What is allotype? You have to remember. Allotype means different allelic form or simply polymorphism. What is allele of or allotype of antibody? Let's assume that I have already mentioned this, the antibody composed of heavy and light chain. There are some antibody or some heavy and light chain that is formed in different allele type. Let's assume this is H1, L1. There are also heavy chain like H2, L2. So these are the allelic form, different allelic form of a heavy chain and light chain. Let's assume this is a IgG antibody heavy chain and light chain. this gene segment H1 and L1 can be present in two different allelic form, H1, H2, L1, L2. So these are called allelic form or polymorphism or allotype. Okay, let's assume this is same species, like species A and a species A. This is a two same species but two different individual. This is A1 and A2, same species but different individual. If we transfer antibody from these two different individual, they will develop anti-allotype antibody. Let's assume this IgG antibody composed of heavy and light chain. Maybe this A1 individual have H1 L1 type of antibody IgG, and this A2 consists of IgG but H2 L2 variant. And if we transfer the antibody, then the individual will develop anti-allotype antibody. So in this case, the anti-allotype antibody also bind with constant region of heavy and light chain. This anti-allotype antibody bind with constant region, C region of heavy and light chain of different allelic form, and it is generated if we transfer antibody in a single species but different individual. You have to. The next is anti-idiotype antibody. So this antibody is also against, but it is against on idiotype. So you have to remember what is idiotype. Idiotype means binding region of antigen of a antibody. The region of antibody that can bind with antigen, that called idiotype. And we know that the variable region of antibody, this variable region of antibody is responsible for antigen binding. This is called FAB, fragment of antigen binding, and the rest is called FC, means fragment of crystallizable. The idiotype, it will not bind the constant region as for the anti-allotype and anti-isotype antibody, but this idiotype antibody bind with this region or antigen binding region. Okay, there is a difference. If we transfer antibody between identical twin, then the anti-idiotype antibody can be generated, and this antibody bind with FAB region, means antigen binding region, or you can very specifically call this CDR. CDR means complementary determining region. Complementary determining region. In the both this variable region of heavy and light chain, there are this region called hypervariable region. In the variable region, there is these three stages, short stages, approximately 10 amino acid. They are composed of this 3 and 3, 6 loop, actually called hypervariable region, because most variability remains in this portion of the variable domain of heavy chain and light chain. These three stretches called CDR1, CDR2 and CDR3. So this CDR means this hypervariable region of heavy and light chain. So anti-idiotype antibody bind in this region,